Hello, and welcome to our online lecture for Psych 1101 and Psych 1010 at Lanier Technical College. My name is Michael Holman. I am a psychology instructor here at Lanier Tech, and I will be your narrator. Please note that these lectures are intended to assist you in better understanding the material, and should not be considered a substitute for attending lecture, reading the text, or completing your assignment. So, this week we're going to talk about sensation and perception. We are going to define and differentiate between sensation and perception. We will identify the parts of the eye, describe the properties of light, and the theories of color vision. We will describe how visual perception is organized, identify the parts of the ear, and explain the sense of hearing. We will describe the chemical senses, identify the skin senses and theoretical explanations for pain. We will describe the kinesthetic and vestibular senses, and explain why psychologists are skeptical about extrasensory perception, or ESP. So true or false? People have 5 senses. That is false. If we could see waves of light with slightly longer wavelengths, warm-blooded animals would glow in the dark. That is true. When we mix blue and yellow light, we obtain green light. That is false. Many people experience pain in limbs that have been amputated. That is true. Some people can read other people's minds. As far as we know, that is false. So, sensation and perception. Well, to start with, we have to define the two terms separately. Sensation is stimulation of sensory receptors and transmission of sensory information to the central nervous system. It is information coming into your senses. It is stuff coming from your eyeballs to your brain. That is sensation. Perception is the process by which sensations are organized and interpreted to form an inner representation of the world. So if sensation is information that comes in through your sensory organs, perception is what your brain does with all that information. So it's how you organize it. When perceptions don't accurately represent the world, they're known as illusions. So this is when you see something, but it's not representative of reality. For example, when you look at this black box with the line, you probably see the black line going through it, right? However, that's not the case. You see a line going through it, but if we back up a second, that line isn't here in the black box. This is a black line, this is a black box, and then this is a black line. But the line can't go through it because they're the same color. Our brains automatically fill in the gaps, complete the space between the lines to assume that it is one line. This one you guys have probably seen before in a previous video. Which center circle is larger? Well, you're hopefully aware of my tricks by now, but if you take away the outside circles for either center circle, you see that they are actually the same size. The added context of the larger circles for this top one and the smaller circles for this bottom one made them look larger and smaller. This is part of a field of psychology known as psychophysics, which studies the relationship between physical events and the corresponding experience of those events. Psychophysics looks at things like thresholds, such as the absolute threshold, the J&D, or just noticeable difference, Weber's law, all sorts of stuff. So let's start with the absolute threshold. The absolute threshold is the weakest amount of a stimulus that can be distinguished from no stimulus at all. It is detected 50% of the time. So this is the smallest amount that you need of any stimulus, whether it's light or sound or smell or whatever, to actually detect it, to notice that it's there. So if absolute threshold is the smallest quantity of physical energy that can be reliably detected, with our eyes, the visible spectrum is within our absolute threshold. But notice that there are lots of other wavelengths of light floating around, all around us, in space, in the air, everywhere. You got radio waves, TV waves, microwaves, infrared waves, and the visible spectrum is just a little bit below infrared waves, but it is a little bit above ultraviolet waves or UV waves, what you might see from, getting a sunburn or something like that. So these are the bits of light that we can see, but there's all these others that our eyes simply aren't designed to see. So here are just some examples of the absolute sensory threshold so that you can get a good idea. With vision, imagine that you could see a single candle flame from 30 miles away on a dark, clear night. Hearing, you might hear the tick of a watch from 20 feet in total quiet. Smell, you might have one drop of perfume in a 6-room apartment. I always will tell students if it's like a really strong cologne or something like Axe or one of those awful bathroom colognes like Sex Panther or something, it just takes one drop and then you just smell it throughout the whole house, right? That's an absolute threshold. Touch, feeling the very smallest amount. It says the wing of a bee on your cheek dropped from 1 centimeter. Another example might be if you're laying in bed and you feel something on your thigh. You don't know what it is, but you feel something. You can just tell that there's something. And then you find it's a blade of grass, a single blade of grass that is just touching your leg while you were trying to sleep. That's an example of an absolute sensory threshold. And taste. 1 teaspoon of sugar in 2 gallons of water. Some people, if you even think about adding certain spice to pasta sauces or something like that, they can taste it, right? That's what we mean when we're talking about absolute sensory threshold. The smallest bare minimum amount before you can tell that something is there. In a similar vein as absolute threshold is the difference threshold. Now, instead of the minimum amount that we need to detect something at all, we're looking for the minimum difference between 2 stimuli in order to tell them apart. So I could have 2 sounds playing at the same time and at the same pitch, but one of them is just slightly off. That's the difference threshold, and you only notice it about half the time. Weber's law, or Weber's constant, is saying that there must be a standard of difference, and this standard of difference is called the JND, the just noticeable difference, which is the minimum difference a person can detect. And it works off of the same principle as the absolute threshold, so it's just the very smallest amount of difference that you can tell between 2 colors, or between 2 sources of light, or 2 sounds, or 2 smells, anything like that. Signal detection theory is a theory that states that stimulus characteristics and psychological factors interact to influence whether a stimulus is detected. So you might have learned to look for specific colors. You might be motivated. For example, you might be less likely to find Waldo in a Where's Waldo book if you don't know that you're looking at a Where's Waldo book. But if you're trying to find him, then suddenly he becomes more apparent because you were motivated to find him. And also psychological states. You'll see this a lot where our mental state impacts what we see and what we don't see. For example, when we're afraid, we tend to get tunnel vision, and we only see the thing that we're afraid of. When we're really happy, we tend to see all the details in the things around us. So our mental states absolutely can affect what we perceive. Signal detection theory likes to test their idea by doing a little exam, you might say. And in this exam, what they'll do is they'll get one of these old devices. They're ancient, you might have heard of them. They're called CD players. And they will have a participant listen to the CD. And the CD is just a recording of people making noise in like a cafeteria. And it starts out with no noise, and then one person talking, and then a couple people, and a couple people, and a couple people, and before you know it, it sounds like a whole stadium is in there just making tons of noise. And your job as the listener is to listen for a beep the entire time. So you just have to tell us after each track, do you hear the beep? Your sensitivity is the threshold level needed for distinguishing signal from the noise. Keep in mind, we don't always send that signal in the noise. Sometimes there is no signal there. And that is where bias comes in. Many times participants will be too willing to report that they heard the beep even though we didn't send a beep. So, in those cases where we either did send a signal or we didn't, and the participant either reported hearing a signal or they didn't, we have a few designations. If we did send a signal and the participant said that they heard a signal, then it is what we call a hit. Means they correctly identified the signal in the noise. If we sent a signal and they said they didn't hear it, then they missed it. Simple as that. If we did not send a signal and they say they heard something, it is called a false alarm. And if we did not send anything and they said they didn't hear anything, it is known as a correct rejection. This brings us to feature detectors. Now, hopefully you remember from our last lecture where we talked about neurons and how there were sensory neurons and things like that. Well, that's coming back into play today. Feature detectors are those sensory neurons. So neurons respond to different aspects of different features. So you've got special neurons for your eyeballs, special neurons for your ears, for your nose, all sorts of things. So there are special sensory neurons for each of your senses. Sensitization is part of something known as sensory adaptation, or your senses adapting to your environment so you're not overwhelmed. Sensitization is part of something called positive adaptation. This is the process by which we become more sensitive to stimuli of low magnitude. So, going into a dark room, your eyes will eventually become more sensitive to light so that you can try and see. Desensitization is the opposite. It is negative adaptation. Now remember, this is something that you're going to want to write down. In psychology, 90% of the time, positive means adding something to the situation. Negative means taking something away from the situation. It does not mean good and bad 90% of the time. It means the aforementioned. So, positive adaptation, you added sensitivity. Desensitization, negative adaptation, you took sensitivity away. So this would be if you came out of a dark room and now your eyes are adjusting, right? They're becoming desensitized. They're not as sensitive to all that light. Speaking of light, let's start with vision. So vision's bread and butter is light. This is a spectrum of electromagnetic energy. We've talked about it a little bit already. Light can vary in wavelength. Within light that we can see, or visible light, color is determined by this wavelength, and a prism separates wavelengths. So this is the visible spectrum. And as I've mentioned before, ultraviolet rays and gamma rays, X-rays, all that stuff is stuff that we can't see. They are just not wavelengths that we can see. It's too low. And infrared rays through radar and FM radio waves, TV waves, AM radio waves, and AC circuits, all that stuff is also floating in the air, but their wavelengths are too high, so we can't see them either. Here's another thing that you want to keep in mind when we're talking about, well, a lot of things. Remember we keep talking about wavelengths? Well, there are a few definitions that you want to remember. So this graph is really helpful for remembering these wavelengths because they'll come up again and again and again. Within a wave, you have certain physical properties. And this is good for light waves, sound waves, brain waves, any sort. They all have the same kind of properties. There will be a wavelength, a wave amplitude. The length is how long is the wave. As you can see here, this is the length of this wave. From this one to this one. So between waves, how is it? And the wave amplitude is the height. So wavelength is the length. Wave amplitude is the wave height. For light, the wavelength is how we determine the hue or the color. For light, the wave amplitude is how we determine its brightness. And in this case, we also have purity, which is how saturated that light is. So as we've already said, we have amplitude, we have wavelength, we also have frequency. This is another major aspect of waves in general. Frequency just means how many waves am I looking at in this sequence? Is it a lot? Is it only a couple? What is the amount? With the eye, which is our sensory organ that deals with light, light enters through a narrow opening. We have the cornea, which is the transparent eye cover, the iris, the pupil, all sorts of stuff. And you can pause this if you'd like to write down these definitions. We have your lens, which can change in thickness to allow you to focus, and an image is then projected onto your retina. Your retina is the light-sensitive surface, and this is where those special sensory neurons are hanging out. You've got photoreceptors, such as rods, cones, bipolar and ganglion cells. All these guys are hanging out, and they receive the light and then send it to your brain. They send it through something called the optic nerve. The optic nerve is the main, it is the only pipeline from your eyeballs to your brain. If this nerve were severed or damaged, then no matter how well your eyeballs worked, you wouldn't be able to see because your brain would never get that information. So the axons, hopefully that's familiar, of ganglion neurons will form the optic nerve and then conduct sensory input to your brain all the way to your occipital lobe. Now, this is really a very handy chart for memorizing what the different parts of the eyeball are and what they're called and what they do, that sort of stuff. So we're going to start on the outside here with the cornea. The cornea is this white part. It is the covering of your eye. It is just the protective covering. So the way that I always get students to remember this is through an awful story. So when I was in school, we had this girl, and had classes with her all the time. And one day, she's in the bathroom, and when she comes out, she's rubbing her eye. And she just keeps rubbing it, and we're like, what are you doing? And she's like, well, my contact rolled behind my eye, and I'm trying to get to it. And so she was trying to get to it, and she kept working on it. And then she goes, oh, I think I got it, I think I got it. And as she was pulling and pulling, she was not pulling on her contact. She was pulling on a piece of her eyeball, on the white. She was pulling on the cornea. Now, take a moment to, collect yourself, because I know you're probably wincing at that story, but that shows the entire purpose of the cornea. The cornea is there to protect our eyeball from dust, from dirt, from air, and yes, from fingers. So that is its whole job, and it did good because she's not blind today, as far as I know. Moving on, we have the iris. The iris is the colored part of your eye. So if you have blue eyes or brown eyes, that color is your iris. The iris expands and contracts. And when it expands and contracts, it makes the hole at the center of it bigger or smaller. And that hole is your pupil. The pupil is where light enters your eyeball. So if you go to the eye doctor and they put that stuff in your eyes and now your pupils are really dilated, what they've done is they've made your iris enlarge, as big as it can get, so that the most light can enter your eyes so that they can better examine you. Now, after light has entered your pupil, it hits the lenses on the inside of your eyeball. And this works exactly like glasses. Glasses are just manufactured lenses. And so light hits this lens, and then the image is bounced off of the lens and onto your retina. And the retina has all sorts of rods and cones and sends the stuff through the optic nerve on into your brain. So cones are most densely packed in the center of your retina, which is called the fovea. Cones provide color vision. An easy way to remember this: cones, color. They both start with C. Also, if that doesn't work, traffic cones are orange. It's a color. Rods provide vision in black and white, and they're more sensitive to dim light than cones, and they are in the rest of the retina, so they're all around. So rods are black and white, cones are color. So we've got about 100 to 120 million rods in our eyeballs at any time. And these rods are very sensitive to light, but they only register shades of gray when sending it to the brain. They are not in the fovea, they're on the rest of the retina. Cones, we've only got about 5 to 6 million. These guys are sensitive to particular wavelengths and produce color vision. They are also much denser near the fovea, or the center of the retina. Your visual acuity, or how good you can see, really, is greatest in your fovea. Makes sense, that's where most of your cones are. But we have a blind spot. This is a point in your retina where ganglion cells converge. Here's a fun way to figure out your blind spot. Take both of your fingers, your pointer fingers, and bring them in front of your face. Look at them. Now, staring straight ahead, do not move your eyes. Move your fingers all the way to the sides of your head. Stretch out your arms and move your fingers to the side of your head, and stop when you can no longer see both your fingers on either side of your head, making sure that you're looking directly forward. That place where you cannot see your fingers, that is your blind spot. This is why we have mirrors in cars. You can put your hands down now. So this is why we have rear view mirrors, why we have those driver's side mirrors, all those sorts of things. It's because, sure, we could look to our left, we could look to our right, probably still a good idea, but we need those mirrors because somebody could be in our blind spot, and you want to check when you're driving around on the interstate to make sure that no other car is there, or you're going to get in an accident. So that is why. And the reason why that we have those blind spots is because that is where the points in the retina are converging to create that optic nerve to send off to the brain. So where that information is going to the brain, they're obviously not converting information. So that's why you have that blind spot there. Now, in some people, they have nearsightedness. Other people have farsightedness. Nearsightedness is exactly what it sounds like. You can see close up, but you have trouble seeing far away. Farsightedness, you can see far away, but you have trouble seeing close up. Presbyopia is a condition that affects many adults, myself included. This is when your eyes were fine when you were young, but as you've gotten older, your vision has declined. So it has gotten worse with age. For example, I used to have 20/20 vision. Didn't matter where I was or who I was with, I could just see everywhere, you know? No problem, could see at night, anytime. Now, I gotta wear glasses. I have trouble seeing things in the distance. I am nearly blind at night, and this is all part of presbyopia. So it's just a condition in which your eyes start to decline as you get older. Now, we mentioned this a little bit before, but let's talk about light adaptation. We have to start with dark adaptation then, the process of adjusting to being in lower lighting. What we know is that your cones reach maximum adaptation in about 10 minutes, but your rods will continue to adapt up to 45 minutes. So if you ever find yourself in an incredibly dark area like the woods or a dark room, a basement, something like that, and you want to see how long it takes for you to fully be able to see in the dark, no, it's going to take about 10 minutes and you'll see all the color that you're going to see. But your rods will continue to adapt for up to 45 minutes. So by 45 minutes, you will be fully adapted to being in the darkness, and you will see as much detail as you can after 45 minutes. However, adapting to bright light is a lot faster. So you come out of that dark room, and now you're in a bright room, and your eyes adjust completely within a minute or so. And honestly, this is a good thing, because imagine coming out of a dark room every morning when you wake up, and then, ouch, my eyes, and then it takes 45 minutes before your eyes can finally adjust. That would be awful, right? So it's a good thing that we're able to adapt to more light so quickly. Now, the visual process works like this. It starts with transduction. Waves of light don't just go directly to our brains. Instead, what happens is they hit our retina, they go through the rods, they go through the cones, and then something known as transduction occurs. And this is very important. Transduction is the conversion, the transformation of light waves into electrical energy. Remember, your brain works on neural impulses or electrochemical signals, right? So transduction is taking light and transforming it into an electrical signal that is then sent to your brain. That's what transduction is. Accommodation is when our bodies are able to accommodate to this new information. Things like dark adaptation is accommodating to a new situation. So you're changing depending on the situation. And of course, we already know about blind spots or filling in the blanks. That's why we might turn our head when merging lanes. We've talked about this a little bit already as well. We've got hue, which is the color of a wavelength of light. We've got value, which is the degree of how bright or dark that wave of light is, or that ray of light, really. And saturation, how intense is the color for that light. When understanding dimensions of color, we have to talk about the color wheel. This bends the colors of the spectrum into a circle so that it's easier for us to really describe how different colors of light interact with one another. So we call the colors on a color wheel either warm or cool colors. If you are on the green or blue side of the color wheel, you're cool. Yellow, orange, or red side, you're considered warm. Makes sense. So this is the famous color wheel. And as you can see, anything on the reddish side is considered more warm. Anything on the bluer side is considered more cool. Notice the gray that's in the middle. That'll be important in a couple slides. So across from each other in the color wheel, we have what are known as complementary colors. So these are across each other in the color wheel, and when you mix complementary colors, you actually get gray. Remember, we're talking about colors of light, so different rays of light that are different colors. We're not talking about pigments, so not, combining red paint with blue paint and now you have purple. That makes sense for pigments, that's in paint, but it doesn't make sense for rays of light. So you got to get that out of your head. That's something different. Remember that light is the source of all colors, and pigments simply reflect and absorb light selectively. For example, did you know that the color white is not actually a color? It is all color. It is all light. What happens is rays of light hit, say, a white shirt, and the pigments on that shirt bounce off every single ray of light. And all of that light then goes back into your eyes. And when you see it, you see all the light, and so it comes out as white. Black is when the pigments in, say, a black shirt absorb all the colors, so it is all colors. So black is when all colors of that light have been absorbed. And so when you see something that is really, really black, it's because that pigment has absorbed everything and you're actually seeing the absence of light in that moment, or in that pigment rather. When you see specific colors, it's because the pigments have absorbed all light except for that particular wavelength. So if you see a red shirt, the pigment in that shirt has absorbed every wavelength of light that has hit it except for the red wavelength. And so that red wavelength is bounced off the pigment, it's rejected, and hit by your eyeballs. So when it gets to your eyes, you see red because all the others were absorbed. Okay, so that was a lot. But with complementary colors, it does make a lot of sense. And now we're going to have a little activity to see how much it makes sense. So, in the following slide, fix your eyes on the dot in the center of the flag. Ready? Let's try it. This works best if you do not blink. Keep staring. Keep going. Keep it up. Do you still see the flag? Most people do, and this is something known as an afterimage. This is the persistent sensation of color, which is followed by the perception of the complementary color when the first color is removed. So this flag was yellow, black, and green, and you probably are still seeing a little bit of that flag right now. And if we go back to the color wheel, you can see that the colors you're probably seeing on the screen, which are slowly fading away, are going to be the opposite of that. So that's just an example of how afterimages work. This brings us to the theories of color. So in psychology, we have many theories about many things, and they all are a little bit right. But we keep testing them, and we keep talking about them and teaching them because they all have good ideas. So let's start with the first theory, trichromatic theory. It says that there are 3 types of cones, and that these cones each respond to either red, green, or blue wavelengths in some way. Another theory says that there are 3 types of color receptors. This is called the opponent process theory. It says that some color receptors respond to red or green wavelengths, others respond to blue or yellow, and others respond to light or dark wavelengths, but that they cannot respond to both simultaneously. So what that means is if you have, say, a red shirt and you have green pants, you might have a color receptor in your eye that can see red and it can see green, but it can't see both the shirt and the pants at the same time. So it needs to respond to one or the other. It will have to have a buddy that can also respond to red or green, and that one will get the slack for the other one that you don't see with that. Now, this theory, opponent process theory, helps to explain things like color blindness. If you are a trichromat, it means that you have normal color vision. All of your rods and cones are working just fine. That's great. If you are a monochromat, it means you are totally colorblind. This is actually a very rare condition, and it basically means none of your cones work. You only see in shades of gray. This is very rare. Most people, even those that are colorblind, are in fact some form of a dichromat. This means they actually have partial color blindness, and that they have trouble discriminating between 2 colors. So they might not be able to see red, they might not be able to see green, something like that. This is more common in males, but we do see it occasionally in females. Here are plates from a test for color blindness. When looking at these plates, you should see the number 6 on the top plate or the number 12 on the bottom plate. If you do not see either of those numbers, you may want to go to a doctor. Eye doctor. So, next, moving on, we've talked a lot about how your eyes work, but now we need to talk about perception. That was the sensation part. Let's talk about how your eyes perceive this information that your eyeballs are sending. How does your brain work with it? Visual perception is the process used to organize sensory impressions caused by the light that strikes our eyes. Sensation is a mechanical process, but perception is an active process. So sensation is just something that we do automatically. It's just light entering our eyes and then it goes to our brain. Boom, done. Don't have to think about it. But perception is something that requires our experience, what we're expecting to see, and motivations. It's our brain. It's how we organize that sensory information coming in. There are many, many rules for perceptual organization, especially with vision. We're going to go over a few of them. The first is something like the figure ground perception. This helps with ambiguous, unstable figures. There are also things like inserting edges. For example, if you look at this image, and if you were to pause this video and go really close, just put your face right up to the screen, you would see that there are actually no edges anywhere in this video. I know it looks like a dock with some sailboats and all sorts of stuff, but the artist purposefully chose not to draw a single line. Every single pixel in this picture is actually just a bunch of little dots. Round, perfectly round dots. So there is not an edge anywhere on this picture, yet we see one. That is the power of our perceptual organization, what our brain chooses to make us see. Here's another example of figure ground. So what figure ground does is it causes us to shift how we perceive things simply because of how close they are to us and how far in the background something is. The Rubin Vase is a great way of showing the power of figure ground. For example, if we look at figure A, you will either see the vase in blue, or you'll see two faces here in yellow. In figure B, though, we've decided to use figure ground. And so we've taken the shape of this vase and we've moved it forward. And we pushed the yellow square to the background. Now, seeing the vase is really easy. But you could still kind of see the yellow faces if you tried hard. Here, we did the same thing, but notice that we brought the yellow square to the front. So now you can more easily see the yellow faces, but if you try a little hard, you can also see the vase in the cutout. That is the power of figure ground. Other rules for perceptual organization come from the school of Gestalt psychology, which hopefully you remember from our first video for the founders and their theories. So some of these rules include proximity, or how close an object is to someone, or to another object rather. Similarity, how similar is that object to another object. Continuity, series of points having unity. So you just assume that they're all connected. For example, with proximity in figure A, how many groups of lines do you see? If you said 3, why? I never told you that there were 3 groups. Why didn't you see 2 groups, Why didn't you see this is one group and this is one group? Well, you know why. Because these two are close together, and these two are close together, and these two are close together, so we automatically group them together. We just say that that's a group, that's a group, that's a group. But there were no instructions telling us that these were groups. We just did it automatically. Similarity. This is our brain telling us that we love patterns. So when you look at this image, you automatically see there are X's all along these rows, O's all along these rows, you see XO, XO, any number of different things. But the point is that you see those similar patterns very quickly. Next we have figure C, continuity. You guys are probably aware of my tricks by now, but let's just see. When you look at this image, do you see a circle with a line running through it? You might, but you shouldn't, because it's really not a circle with a line running through it. Is there a line right here? No, there is not. There is a line coming from here to here, and there is a line from here to here, but there's not a line in that circle. But our brains love to complete things. If they see there's a gap, they automatically fix it. And so we just assume, oh yeah, it's running right through it. Oops, sorry. And this last one, closure. Do you see a circle? If you do, you're wrong, because it's not a circle. It's a bunch of hash marks in the shape of a circle. But we automatically assume that is a circle, because our brains love closure. They love to make sense of everything. And this bites us more than once. Another rule that we'll see is common fate. This is elements moving together, therefore we assume that they're grouped together. Have you ever seen a bunch of birds flying in formation across the sky? And you just assume that they're all flying together, right? But you know, it's not like those birds got together and had a meeting and said, okay, James, I need you to take point, and Ashley, I'm going to have you on the left, and George, I want you on the right, you know? There was no, okay, we are going to the Bahamas, this is a group effort from the geese flying in the sky. No, we just see them flying, they were just going in the same direction, and we assume, oh, they're all going together. Closure. Closure is the perception of a complete figure even when there are gaps in sensory information. We saw this with the dotted line that looked like a circle. Here's some more ambiguous figures. You've already seen the Rubin Vase, but what about the lady on the right? Do you see a young woman, or do you see an old woman? Many people will see either one or the other. But once you learn the secret to how you can see either one, then you can switch back and forth. Feel free to pause this video if you are struggling to see the old woman or the young woman. Easy ways to figure it out. If you see the young woman, then imagine that her chin is a nose, a giant nose, and the young woman's ear is actually an eye, and her necklace is actually a mouth. That should help you to see an old woman. If you see the old woman, then it's the opposite. Imagine that her mouth is a necklace, and her nose is actually part of her chin, and going up her jaw, and her eye is actually her ear. What's great about this particular figure is that once you see both the old woman and the young woman, you can go back and forth. And that is the amazing power of perception. We are all sensing the same thing. We're all getting the same sensation. But you will see different things depending on what you tell your brain to allow you to perceive. That's the power of perception. Another important aspect of perception is top-down versus bottom-up processing. Top-down processing is perception of the whole followed by perception of the parts. So this is when you recognize something. Bottom-up processing is when you have perception of the parts that leads to perception of the whole. So if you have never seen a car before in your entire life, that's bottom-up processing. The first time you see it, you go, oh my god, what is that thing? It's got doors, and it's got a windshield, and it's got 4 wheels. It's got a grill. What is this? And then someone says, oh, that's an automobile, right? So the next time you see a car, you go, oh, that's a car. The first time you saw it, you were building up your idea of what this was from the bottom up, from the ground up. But the second time you saw it, you recognized it. So now you're looking at it from the top down. It's a way to remember it. So we have top-down processing and bottom-up processing, but we also have perceptual sets. And this is how top-down processing can get us into trouble. For example, what does this word say? If you said beer, you would be wrong. But why did you say beer in the first place? Because this looks like a B, that looks like an E, that looks like an E, and that looks like an R. Oh, that makes the word beer. That makes sense. Except the E and the F look exactly the same. So perceptual sets can get us into a little bit of trouble every now and then. It's when we make these assumptions. We also can perceive motion. The visual perception of motion is based on the change of position relative to other objects. And when we change this position, we can create the illusion of movement. This is something known as stroboscopic motion. And this is how films, cartoons, animations of any kind, movies, TV shows, that's how they all work. It's just a bunch of still images played together rapidly, creating the illusion that they're moving. Another important aspect of visual perception is depth perception, and this relies on monocular cues. So monocular cues, mono as in one. Think of it like a monocle. So This is a little eyeglass that only goes on one eye. So these cues only happen in one of your eyes. We have perspective, clearness, overlapping, shadows, texture gradient, and motion parallax. Binocular cues, bi as in two, or binoculars, which require two eyes. So these are cues that require both of your eyes to work together in order to perceive that depth. So this is things like retinal disparity, meaning that one eye sees one thing differently than the other, and so you compare notes, and convergence, when both eyes look at the same object and combine the different aspects that they're seeing. Perceptual constancies are acquired through experience and create stability in our lives. Things like size constancy, color constancy, brightness constancy, and shape constancy. It is knowing, for example, that a pencil is always going to be the size of a pencil. Go ahead and take up a pencil or a pen and stare at it from afar. Hold it in the air and look at it. Take note of the size of the pencil. Now, bring it closer and closer and closer and closer to your face. Why aren't you freaking out? Why aren't you thinking that, oh my god, this pencil has gotten gigantic? Well, that's because of size constancy. You know that a pencil stays the size of a pencil no matter how big it appears in that moment. It's the same principle with color constancy, brightness constancy, and shape constancy. For example, even if I were to shift these cubes over, we still know that they have the same size, the same shape, and the same color, even if they look a little bit different. One of the great things about this chapter is that we get to talk about illusion, things like the Herring-Helmholtz illusion, where you might perceive a drawing as being three-dimensional, or the Mueller-Lyer illusion, where you interpret the length of lines based on experience. Here's an example of the Herring-Helmholtz illusion. It looks like a tunnel, right? Looks very three-dimensional, but clearly this is just a 2D image. The Mueller-Lyer illusion is very cool. So this is where we can compare these two lines right next to each other, and as you can see, they're the same length. Yep, same length, they just have different arrows. And yet, when I move these arrows over here, they look drastically different, don't they? You look like you have an entirely different angle, and that this is a shorter wall and this is a longer wall. But we know that they're the same. This is just copy and pasted from over here. That's the Mueller-Lyer illusion. So what you see is not necessarily what you're getting. So how does visual perception work? Well, it starts with pathways. It starts from your eyeballs, goes through the occipital lobe, and then from there, figuring out where the thing you're looking at is goes to your parietal lobes, and figuring out what the thing you're looking at is goes to your temporal lobes. Here's another activity for you guys. On the next slide, I want you to locate the red T. Are you ready? You probably found it, right? On the next slide, locate the red T again. Probably took a minute, but you found it, I'm sure. On the next slide, name the colors of the ink as quickly as possible. This is pretty simple, right? It's red, it's blue, it's pink, it's green, it's red, it's green, it's blue, and on and on and on. On the next slide, name the colors of the ink as quickly as possible. Again, not so bad, a little tricky, but not so bad. Now let's try dividing your attention. On the next slide, name the colors of the ink as quickly as possible. Take your time. This one is not so easy, right? Because you're reading a word, and you want to say the word that you're reading, but instead, you are being asked to say the color of the ink of that word. So you want to say red, but you should say blue. So you should be saying blue, pink, red, red, blue, blue, red, green, green, blue, blue, pink, red, blue, green, pink, red, blue, pink, green, red, pink, blue, blue. Not easy. Now, a lot of times we will see without awareness, and this is part of something known as blind sight. So this is when you just don't see it. You've heard the expression, if it was a snake, it would have bit me, right? That's blind sight. That is, it is right in front of me, and I'm blind to it. I don't see it. I'm just not aware. That is seeing without awareness. You might also be blind, quote unquote, to changes. You could be so focused on something that you don't realize that it's different. There's also repetition blindness. Something could be happening over and over and over again right in front of you, and you don't notice the pattern because you're too focused. Attentional blink is when you missed it because you blinked. You weren't paying attention to that one thing. Take a look at these images. In the first set of images on the left, there is one thing that is different about the images. What is that one difference? Feel free to pause if you need to. The answer is the bar behind them. So this bar is at their shoulders, while here the bar is at their heads. So that's the difference between these two images. Try it again on the right side. There is one thing that is different about these two images. What is it? Feel free to pause if you need to. But you probably got the answer to this one very quickly. This one has the helicopter here, and here the helicopter is much higher in the window. But notice that it was a lot easier for most people to discover the difference between helicopters after they solved this one. Why? Because of experience. Remember that our perception is changed based on experience. Because you've had the experience of the bar, you knew what you could look for when looking at the second image. So that's another example of how perception changes just like that. So that was enough about vision. We're not talking about vision anymore. Now we're going to focus on hearing. Hearing's bread and butter is sound. Just like light works for eyeballs, your ears work based off of sound. Sound waves require a medium like air or water and compress and expand molecules of the medium, creating vibrations. A single cycle of compression and expansion is one wave of sound. The human ear is sensitive to sound waves with frequencies of 20 to 20,000 cycles per second. Pitch is the frequency. Remember that? So that's the frequency of the sound waves, number of cycles per second. It is expressed in hertz, and for example, the pitch of a woman's voice is higher than men's. Loudness is the amplitude, or the height, of sound waves, and is expressed in decibels. So once again, we've got various frequencies and amplitudes, right? And those tell us all sorts of information, but now we're talking about sound waves. But it's the same principle that we talked about with light waves. Here are just some interesting ratings for decibels and how they can get dangerous. So, for example, at 20 decibels, it's considered very quiet. This is a broadcast studio when it's quiet, whispers at 5 feet. Moving all the way up to a normal conversation at 60 decibels. Your average car is 70. Things start to get very loud after 80 decibels. Factory noises, chainsaws are about 110. Thunder, rock concert, basketball, or hockey crowd is just below 130. You are in immediate danger at this point, and it's considered extremely loud. At this point, you've reached your threshold for pain. So that's just past 130. This is having a stereo headset at full volume, hearing a siren at 50 feet, a jet airplane, a shotgun blast. Anything past 160, hearing loss is certain. And if you made it all the way up to 180 decibels, which would be equivalent to a sound of a space shuttle launching, you're probably going to die, just from the sound alone. So the ear is shaped and structured to capture sound waves, which vibrate in sympathy with them and transmit auditory information to the brain. So here's a human ear. You got your outer ear, which is the auditory canal. This is the part of the ear that we're all familiar with. And sound waves will enter the outer ear, hit your eardrum, and vibrate it. That's creating that energy. This eardrum then pulses, which causes these three bones in your ears, the hammer, anvil, and stirrup, to smash together. Then, this energy created by these bones is sent into the cochlea, and the cochlea is really where the magic happens. So we have your outer ear, your bones, which are part of your middle ear, and then your inner ear, which is part of your cochlea and your auditory nerve. So outer ear funnels sound waves to the eardrum. The middle ear is your eardrum to your hammer, anvil, and stirrup. They act as amplifiers and send through an oval window into the inner ear, which is composed of the cochlea and the auditory nerve. The cochlea has a basilar membrane and the organ of Corti. We'll talk about them in a second. So, when we try to locate sounds, the loudness of that sound and the sequence in which the sounds reach the ear can provide cues. So we figure out how loud it is and where it's coming from based on this information. Sometimes you might even turn your head to figure out where the sound is or how to clear up what someone said. We perceive this loudness and this pitch related to the number of receptor neurons that are on our organ of Corti. Sounds are perceived as louder when more of these neurons fire. There are two theories when it comes to understanding how our brains sense loudness and pitch. They are the place theory and the frequency theory. So the place theory says that pitch is sensed according to the place that vibrates. So depending on where in the organ of Corti, this little guy right here, vibrates, that is what your pitch is. The frequency theory says that the pitch is perceived on stimulation of impulses that match the frequency of the sound. So it says that depending on the frequency at which the sound hit this organ of Corti, that's how you tell pitch. And what's most likely the correct answer is that both theories are correct. That different pitches occur depending on where the pitch hits and at what frequency the pitch is hitting in that location. And that's how you can tell different pitches. But when talking about hearing, we have to talk about deafness. And there are two types of deafness. There is conductive deafness, which is damage to the middle ear, and hearing aids can help in this case. And there is sensory neural deafness, so damage to the inner ear or the auditory nerve. If you only have damage to the inner ear, then we can give you a cochlear implant, which can help. But if you have damage to the auditory nerve, it's just like the nerve to your eyes, right? Gone. That is the only pipeline to get to your brain. So if that auditory nerve is damaged, you are deaf. There's nothing that we can do for you. Now, when we organize the auditory world, we do it based on a speech segmentation problem. This is when you hear half of a conversation and you're trying to figure out what somebody actually said. Usually, when you try to figure out what somebody said, you do it based off of categorical perception. So this is when you heard someone talking and you know that what they said doesn't make any sense, right? You just filled in the gaps and you said, I swear I heard you just say that you want to eat a tuna sandwich, but I know that you're a vegetarian. That doesn't make any sense, right? So that is part of categorical perception. You heard something and your brain just filled in the gaps to try and make sense of it. Your brain loves to fill in the gaps. It can get us into a lot of trouble. When we are locating sounds, we use both ears. So you walk along and maybe you hear somebody talking in the hallway and it is really juicy gossip, right? So you are going to pause and then you're going to listen and you're going to try and figure out, where is that sound coming from? Is it coming from my left ear or my right ear? So you decide it's your right ear. So you might hold a hand over your left ear and close it off so that you don't hear sounds coming into your left ear. Now sound is only coming in through your right ear. You might even put your hand up to your right ear to better hear it. Or maybe you take a glass and you put it on the wall so you can really get all the good detail because you're that super nosy neighbor. We notice the difference in loudness, and that's how we can figure out where it's coming from based on our two ears. And that's how we use one ear once we've narrowed it down. This leads us to something called the cocktail party phenomenon. So this is when, again, you hear parts of things, but even though you're in an incredibly loud room, maybe a cocktail party, for example, somebody could say certain things and you will hear it across that room. So you could be talking and it's loud and nothing happens, and somebody happens to mention your name. Boom. You hear it. You hear it across the entire crowd just because you heard your name. So you were able to hear something despite all that extra noise. This is part of something called dichotic listening. Di as in your two ears. Okay, so that's enough of our hearing senses. Let's talk about our chemical senses, which is going to cover two of them. This is smell and taste. So with smell, odors trigger receptor neurons in the olfactory membrane, and odors are sample molecules of substances in the air. Sensory information about odors is sent to the brain through the olfactory nerve, and the odor contributes to the flavor of foods. The olfactory system is a part of your chemical senses, as we've already discussed. So the way that it works is that you have an object, say a nice flower, and molecules float in the wind. They float in the air from that nice flower. So these are pieces of the flower. And those molecules go up into your nose. And they float around in your nose and eventually fly all the way up to the mucus layer. And from the mucus layer, they hit the cell body of olfactory receptors. These are those sensory neurons. And those sensory neurons then send information to your olfactory bulb, which sends it to the brain. These receptors function based on a lock and key mechanism. What this means is that certain smells are only triggered based on certain molecules. So that's why you might say, oh, this smells like it's sweet. This smells really terrible, right? Well, that's because you have a specific lock for that nerve, for that receptor, and it will only unlock when that particular molecule has entered into it. So when that key has entered, then the signal is sent to your brain. I want to talk about pheromones for a second. So you've probably heard of pheromones before. You know, they say that a pheromone is what causes you to be attracted to someone. There is an entire industry built on this, of colognes, of perfumes, and how they make you smell more attractive. Well, that actually does work. That's why the industry is there. But what's interesting with pheromones is that we've actually seen that depending on what you're attracted to, pheromones will work. For example, there was a study done several years ago where we had men that identified as heterosexual and men that identified as homosexual. And we also had women that identified as heterosexual and women that identified as homosexual. When we sprayed perfume and we monitored their brain activity, men that identified as heterosexual, certain areas of their brain lit up. And when men that identified as homosexual smelled this perfume, those parts of their brain did not light up. Similarly, straight women, their parts of the brain did not light up, but homosexual women, when they smelled this pheromone, when they smelled the perfume, the same part of their brain lit up as the men. And in another similarity, when we sprayed cologne, there was really no reaction from straight men or from gay women, but among gay men and straight women, the same areas of the brain lit up. So that was very interesting. The next chemical sense we're going to talk about is taste. Taste is sensed through your taste cells. Receptor neurons are on taste buds, and it's pretty simple. The molecules from your food hits your tongue and then hits the receptor neurons. We know that there are 4 primary taste qualities. Sweet, sour, salty, and bitter. But there's also a fifth, or umami. This is a savory taste. Not everybody has the taste of umami, but those that do, we often will call them super tasters, because they can taste food at a much more powerful rate. So food just tastes more to them. They have different flavor combinations. Flavor of food depends on odor, it depends on texture, it depends on the temperature of the food and taste. And everybody has different taste sensitivities. Some people love it sweet, some people love it bitter, some people love it sour. It just depends on the individual. The top image is a photograph of your tongue surface, magnified about 75 times. There are about 10,000 taste buds that line your tongue and mouth. And taste receptors are down inside each of these buds. Children have more taste buds than adults, and the reason why is because we're adults. We burned them off. How many times have you bit your tongue? How many times have you burned your tongue or gotten it too cold or anything like that? When you did that, you hurt your taste receptors. So kids have more because they haven't had a chance to hurt them yet. Take note of this image. There are little hairs, yes, we have a hairy tongue, that are right above the pore in order for molecules to be tasted. So molecules hit the hair and are then hit by nerve fibers just below in the taste bud. And then they are sent directly from the taste bud to your brain so that you know what you just tasted. Here's another example. As you can see, molecules enter through this pore past the surface of the tongue. They hit receptor cells, which then hits a sensory nerve fiber. On the right, we have an old image of how our tongue senses different tastes. So sour is in the back, bitter is in the very back, salty here, sweet and fatty. But what's interesting is that this is actually incorrect. In fact, we have taste buds that taste everything all over the place. So this is just an example of how we used to think about it. Alright, next we have your skin senses. Skin senses include touch and pressure. So when somebody touches you, when you feel pressure, your skin sensory receptors, those are neurons, remember, fire whenever skin surface is touched, especially during active touching. Some areas of the body are more sensitive than others, and this is where nerve endings are more densely packed. More sensory cortex is devoted to perception of sensations in those areas. So if you have a particularly tender spot on your body, if you've ever gotten tattoos, and you'll know that there are certain areas of the body that are more sensitive than others, that's because there are more nerve endings there. And so it hurts more because there's more nerves telling your brain it hurts. For temperature, receptors are located just beneath the skin. Skin temperature increases, and therefore receptors for warmth fire, but if your skin temperature decreases, receptors for cold fire. So what's interesting is that it's not that you get cold, it's not that you feel cold, and therefore you are cold, it's that your body lowers to a certain temperature degree, and then the receptors in your skin fire, telling you that you're cold. And if your body gets to a certain temperature degree higher, those temperature receptors will fire and tell you that you're warm. But sensations for temperature are relative, and this is why some people are hot-natured and some people are cold-natured. It's because everybody's receptors fire at different degrees. So it could be 80 degrees for one person and they feel great, and another person, they are sweating to death. It's simply because their receptors fire at different temperatures. Pain has special receptors called nociceptors, and when these guys are stimulated in your skin, it's done where nerve endings are densely packed. That is where it is usually sharpest. Pain can even be felt deep within your body, but remember, as we talked about last week, there are no nerve endings for pain in the brain. This is just a handy chart to show you the different sensitivity levels to touch, and you can see that the higher the level, the more sensitive that part of your body is going to be to touch and to pain. Pain runs off of prostaglandins. These facilitate transmission of pain messages and heighten circulation to injured areas. Certain pain-relieving drugs will inhibit the production of these guys, and emotional responses in response to stress can also affect how much pain you feel. Phantom limb pain is something that 2 out of 3 combat veterans with amputated limbs experience. This may involve the activation of nerves in the stump of a missing limb and can also involve reorganization of motor and somatosensory cortex. So phantom limb pain, as mentioned at the very beginning of this video, is when you no longer have that limb, but you still feel pain there. It's because your nerves are still activating and they're trying to sense something. One theory of pain is known as the gate theory. It says that the nervous system can only process a limited amount of stimulation, and that rubbing the pained area competes for neural attention, closing the gate on pain messages to the brain whenever we have too much pain. So gate theory is basically saying that at some point, all right, we get it, you're in pain, this hurts, that's enough. And so it will shut it down, and your brain will no longer accept any further messages of pain. And it does this so that you can eventually get over it. That's also, according to this theory, why rubbing it really does make it feel better. Because it hurts, and you're actually distracting your brain from the pain zone when you rub that area. So experience of pain, according to the gate control theory, depends in part on whether the pain impulse gets past the neurological gate in the spinal cord and thus reaches the brain. Acupuncture is an ancient Chinese method of pain control. Research shows it does stimulate nerves to the hypothalamus, releasing more endorphins, which gives you a feeling of pleasure and feeling better. Endorphins are similar in structure and effect to morphine. Okay, so those were the main 5 senses, but it turns out you got more than 5 senses. So let's talk about kinesthesis and your vestibular sense. Kinesthesis is the sense that informs you about the position and motion of your body. It is your sense of balance. Sensory information is sent to the brain from sensory organs in your joints, your tendons, and your muscles. Your vestibular system is mostly in the semicircular canals of your ears, but it monitors your body's motion and position in relation to gravity. So it helps you stay standing up and walking around and not get dizzy. So that was your 6th sense, but what about ESP, the other 6th sense? Is there really perception without sensation? Are psychics real? Can you really read my mind? What's happening here? Well, ESP, or extrasensory perception, is perception through means other than sensory organs. Things like precognition, which would be seeing the future. Psychokinesis, which would be moving objects with your mind. Telepathy, which is to read someone's mind. And clairvoyance, which is to know that something is happening even though it's not possible for you to know that something is happening in another room or anywhere. So we have studied the existence of ESP using something called the Ganzfeld procedure. This is a method for studying it, but the problem is that there's no reliable evidence for the existence of ESP. So, for example, if you've ever seen the original Ghostbusters movie, then there's a great scene where a man and a woman are being tested by a doctor, and they have these cards with these special symbols on them, and they're supposed to guess what symbol the doctor is looking at. And of course the doctor is a charlatan, and he's been messing with them the entire time. But it turns out that that is, in fact, the Ganzfeld procedure. So if you ever see these little cards, if you watch that movie, know that that's a real thing that we do. However, using that procedure and using many other procedures, we still have not found any evidence to support that ESP exists. So while I can't say definitively that it doesn't, I can tell you that there is no scientific evidence to support that it currently exists. And that is it for this particular video. Please make sure that you're getting all of your assignments submitted by the due date, and I will see you next time for the next lecture.